All right, I'm going to go ahead and get started because I don't want to keep you guys here all day. But I am Mrs. Spencer, and I'll be doing the question review sessions with you guys. Ms. Tyson's doing the content review on Tuesdays, and then Thursdays I'm doing the question review. So how I've set this up is I basically just pulled some sample questions from the EOC item specs that the state provided to us that correlate with the content that Ms. Tyson covered over the Tuesday lesson. And so that way you can kind of get an idea of what some of the question wordings and setups would kind of look like. So we're going to go through, I have 7 different questions that I've pulled, and we're going to go through the question and then talk about why an answer is correct and why the others are not, and then go from there. And if you guys have any questions about any of the material or anything while I'm going through, just let me know. You can type it into the text, the chat box, or you can turn on your mic and talk to me. There's only a couple of you, so it's fine if you want to go ahead and just talk too, okay? So our first question that we're going to look at is this one about osmosis and the movement of water through a cell. So this gives you an idea of what a question might look like. It's got the background information, it's got a chart diagram with some data, and then a question that goes along with it. So I know you guys can read, but I'm just going to go ahead and read it out loud. An osmosis investigation was conducted using 20 chicken eggs to represent cells with semi-permeable membranes. The eggs were first soaked in vinegar to dissolve the shell. The eggs were then transferred to corn syrup for 24 hours and then to distilled water for 24 hours. The mass of each egg was measured before and after each soaking to determine how much water diffused into or out of the cells, or the eggs. This table below shows the average mass of the eggs at each step of the investigation. So we can see that when it was in vinegar, the average mass of the egg before the soaking was 71.2 grams. After soaking in vinegar, it was 98.6 grams. So the difference in the mass was 27.4 grams, and that increased. Then in corn syrup, the egg was 98.6 grams in mass when it started, and then after sitting in that corn syrup for 24 hours, it decreased to 64.5 grams. So that was a difference of 34.1 grams that it went back down. In the distilled water, it started at that 64.5 grams, and then it increased to 105.3 grams, so it had an increase of 40.8 grams in mass. And then this last column is just kind of showing a percent change, okay? So overall, when it was in the vinegar and in the water, the mass increased. When it was in the corn syrup, it decreased. So the question's asking, based on this experiment, which of the following should be inferred about cells with semi-permeable membranes? A, the substances other than water may also cross the cell membrane. B, substances other than water may block pores in the cell membrane. C, water enters the cell when placed in environments of high water concentration. D, water leaves the cell when placed in environments with low concentration of solutes. So what do you guys think this correct answer should be? A, B, C, or D? You can either say it or type it into the text box. C. Rachel says C, and I think our other student has left the class for a minute. So Rachel, that is correct. C is the correct answer, and I'm going to just highlight that real quick here. C. Water enters the cell when placed in environments of high water concentration. And we can see this from this data because the vinegar solution, which was 95% water, and the distilled water solution, which is 100% water, showed huge increases in mass, showing us that water had moved into the cell. D is not correct because this says when they're placed in environments with low concentrations of solutes, the water leaves the cell. So solute is anything dissolved in the water. So if we have a low amount of solute, then that means that there's a high amount of water concentration in the environment. So that would be the vinegar in the distilled water solution. And we can see here that the water obviously didn't leave the cell because the mass increased. So in order for the water to leave the cell, the mass would have had to decrease. And there's not enough information here to tell us to be able to answer A or B. There's not enough information to say one way or the other that substances other than water may cross the cell membrane. We've not given enough. And then B, the same thing, if something might block the pores. There's not enough information in this data that was provided to be able to come to those two conclusions. So we can automatically mark those out. But just to kind of review osmosis and the movement of water through the cell membrane, we can see here in the animal cells, these are red blood cells in the top picture here, and we've got in an isotonic solution, we have an equal amount of water and dissolved substance. Again, remember solute means dissolved substance. So when there's an equal amount in the outside environment versus the inside environment of the cell, it's called isotonic. Iso means the same, and so it's at equilibrium. And so there's not going to be any net change in movement of water across that cell membrane. Whereas here in the middle picture, where B is given, we have very few solutes in the surrounding solution. So there's an excess amount of water. So because of this, the water that's in the outside environment is going to want to rush into the cell to try to equalize that concentration out so that there's an equal amount of water inside and outside. And so that's why the cell swells up and becomes bigger. And that's called a hypotonic solution. I like to remember that hypo with the big O, so it becomes huge and big and round and O, like an O. And then hypertonic is going to be where we have a large number of solutes around the environment of the cell. Very little water is outside of the cell. So that means there's going to be more water inside the cell, so the water is going to try to dilute or diffuse out of the cell to equalize down these solutes. And that's going to cause that cell to shrink up as the water removes itself from there. So we can see this also happens in our plant cells as well. We get a shrinking of the cell happening in the hypertonic. So the first picture here on the left of the plant cell is equal to the C picture of the blood cell. The middle picture on the plant cell is equal to our first A picture on the blood cell. And then the picture here on the left-hand side of the plant cells is equal to our middle B cell of the red blood cell. So it just kind of gives you what it would look like in a plant cell. You don't see as much from the outside of the plant cell because they have that rigid cell wall, but the inside where the vacuole is where you're going to see a huge difference in the shape of the cell. All right. Did you have any questions on that? All right, so we're going to move to the next question. This one's on the cell theory. Okay, good, thanks. All right, the cell theory was first proposed in 1838. Evidence obtained through additional scientific investigations resulted in the current cell theory. So which of those statements describes a component of the original cell theory that was removed because of new scientific knowledge? So which one of those do you think is no longer part of the cell theory currently? Correct. C is false. Excellent. C is the incorrect one. This one was removed from the cell theory because cells do not form through spontaneous generation. That was discovered from Pasteur. He did an experiment that showed that spontaneous generation does not happen, that cells have to come from other pre-existing cells. So just to review that cell theory and the timeline here, the three main parts of the cell theory is that all organisms are made up of one or more cells, and cells are the basic building blocks of life. And this was came to part of the cell theory and came to fruition because of Theodor Schwann and Matthias Schleiden. They both did work with animals and plants. Schwann was with animals, kind of like a swan, that's the way to remember that. Schleiden was with the plants, and their work determined that all of those plants or all of those animals always had cells as a building block and were made up of cells. And then all cells come from existing cells was thanks to Richard Virchow and his work. And then we have to remember that the microscope played a big role in the development of the cell theory, because unless we could see cells, we wouldn't have been able to know any of these things. So the microscope played a big part, and that was developed by a couple different scientists over the years. Janssen was one of the originals, so we had Hooke, who discovered the actual cell itself, and cork cells that he looked at, and then some more microscopes were developed and created over time to become more complex. All right. So next sample question we have, and again, these are actual sample questions from the biology EOC item spec, so similar to how a question might be worded. This one's on the difference between prokaryotic and eukaryotic cells. So there are some similarities between those two types of cells. Which of the following would be something that's found in both the prokaryotic and eukaryotic cell? Which one do you think would be in both of these? No, it's not going to be B. One thing to remember is that eukaryotic cells are going to be our more developed and complex cells. So they're the ones that have the membrane-bound nucleus as well as membrane-bound organelles. So the mitochondria is actually a membrane-bound organelle, so that would only be in the eukaryotic cell. Prokaryotic cells are the early cells that are less complex, so they don't have a defined nucleus and they're not going to have any membrane-bound organelles. So is there something on this list that's not as complex as the others? Correct, ribosomes, very good. The ribosomes are not considered to be a membrane-bound organelle. They are basically blobs of RNA, ultimately, rRNA. So they are found in both prokaryotic cells and eukaryotic cells because they are not considered membrane-bound. Lysosomes is a membrane-bound organelle. It helps with the digestion of waste and things in a cell. Mitochondria, of course, is membrane-bound, helps with energy conversion of glucose into ATP. And then the nucleus is membrane-bound and is only in eukaryotic cells as well. It's the main brain of the cell. Prokaryotes, instead of having a nucleus, will have like a single strand of DNA that is just in the middle of the cell, but it's not actually enclosed in a specific organelle. So the correct answer here would be D. And so I do have a little slide here that kind of shows you comparison between the two, a little Venn diagram. Both prokaryotes and eukaryotes will have DNA. They will both have ribosomes. They both are going to have cytoplasm kind of filling the inside of the cell. And they both have an actual plasma membrane. They both have a cell membrane that, separates the cell from the outside of the cell. The differences are prokaryotes are our oldest type of cell. They're very simple, small. They do not have a nucleus or any other organelles that have membranes. They are single-celled, and the DNA is going to be found in one circular chromosome. Eukaryotes are going to be the more complex cell. They are thought to have evolved from the prokaryote. It's thought that some prokaryotes kind of lived within one another at one point and became dependent so much on one another that that's what developed into the eukaryotic cell. They are going to have a membrane-bound nucleus and the membrane-bound organelles. Eukaryote cells can be found in single-celled organisms or multicellular organisms, and they have multiple chromosomes in that nucleus. So one way to kind of remember, you carry the nucleus, eukaryotes. Eukarry the organelles, eukaryotes. Whereas our pro is the early ones that don't have all that. Okay? Any questions on that? Okay. All right. Next question we have is on division. So we have mitosis and meiosis. Those are processes involved in cellular reproduction. Which of the following describes an event that results from mitosis but not meiosis? So here's an example of a question that's using one of those very important terms, not. So it's very important that you remember to highlight that when you're reading through the question. So here we want to know which one would result from mitosis but not meiosis. So which of these? 2 stages of cell division, replication of cellular genetic material, daughter cells that are identical to the parent cell, or 4 daughter cells being produced. Which of those would not be found in meiosis, but they are in mitosis? Good, yes, C, very good. In mitosis, the daughter cells are identical to the parent cell. Meiosis, on the other hand, we get the 4 daughter cells that are going to be genetically different from the parent cell, because meiosis is involved primarily with making reproductive cells, the sperm and the eggs. Mitosis is used more for growth and repair. It can be used in some asexual reproductions as well, but for multicellular organisms, it's used more for growth and repair. So you're going to want identical cells. So that is why C is the correct answer here. So just to kind of go through a comparison, they both deal with making copies or genetically copying their genetic information prior to the division. I don't believe these diagrams show that, but in interphase before mitosis and before meiosis, all cells will duplicate their genetic information once. Okay? Then they will start the division process. In mitosis, it goes through the prophase, metaphase, anaphase, and telophase once, and then you end up with the 2 separated cells with cytokinesis. So that's where the 2 divisions come into place. Mitosis is the division of the nucleus, and then the cytokinesis is when the actual cytoplasm divides the cell into 2. And you end up with 2 cells, and you can see here these 2 cells on this top diagram at the end look identical to what the parent cell looked like at the beginning. Okay? Oh yeah, they do show where they've duplicated the chromosomes. Good, I was afraid it didn't show that. In meiosis, we get that same duplication that happens, but instead of dividing once, the cell actually divides twice. So we have a cell that divides one time, and then it goes through a division again, so that we end, and that the second division happens without reduplicating the DNA. So there's no second duplication of DNA, it's just a singular, and we end up with the 4 daughter cells that have half of the information that the parent cell had to start with. So that's why they are genetically different, and that's to allow for the combination with the other sex cell to form an organism. So that is the main differences. But they both will have the division of the nucleus, and they both will have cytokinesis, in which the cells literally separate from themselves. And they both will duplicate their genetic information at the beginning of the initial process to start with, okay? Differences is mitosis, we get 2 daughter cells that are identical to themselves as well as the parent. In meiosis, you get 4 daughter cells that are each different from each other as well as from the parent, okay? All right, here's the next question. As food travels through the digestive system, it is exposed to a variety of pH levels. The stomach has a pH of 2 due to the presence of hydrochloric acid. The small intestine has a pH ranging from 7 to 9, which is going to be more basic. HCl converts pepsinogen into pepsin, an enzyme that digests proteins in the stomach. Which of the following most likely happens to the pepsin as it enters the small intestine? So this is a biochemistry question dealing with enzymes and how enzymes are affected. So what do we think might happen to that enzyme once it leaves the stomach at a pH of 2 going to the intestine with a pH of 7 to 9? Very good. Good job, Rachel. A, it becomes inactive. Becomes inactive, okay? And that's because enzymes have very specific conditions that they work in. So it tells us that the pepsin is an enzyme that digests proteins in the stomach. So it's made to work in the stomach with that low pH of 2. So when it leaves the stomach and goes into that higher pH, it's going to become inactive because that's not the conditions that it's made to work in. So that's something to remember here. So there's a lot of fancy words in here, but the main thing is kind of looking at that difference in pH and knowing that it tells you that the particular enzyme is supposed to work in the stomach. So what happens when it moves out of that environment, okay? So just to recap on enzymes, which they are types of proteins, which is one of the macromolecules that's important for life. They enzymes help with chemical reactions, okay? And some factors that will affect how the protein or the enzyme works are pH, which is mentioned in the question we just went over, temperature, and sometimes other chemicals. So when they are exposed to any of these factors that are different than what they are made for, they will become denatured. And denatured means that they don't work properly, so they are going to be, therefore, inactive, like the question said. So if the pH is different than what it's normally used to, if the temperature is too high or too low, or if there's any other type of chemical interference that it's not normally used to for its specific function, then it will become inactive and no longer work. All right? Questions on that? All right, so this question is on photosynthesis and cellular respiration. So the diagram below shows this relationship between photosynthesis and cellular respiration and the organelles in which they occur. So you can see we've got up here the chloroplast with the sunlight energy entering, and then you've got glucose coming off, and then oxygen coming in, and that enters into the mitochondria, which is going to release ATP as well as water. And then if we add carbon dioxide in, we can then go back to it. So it's kind of like a cycled thing here. All right, but you can see with the chloroplast, we have energy from the sun coming in, carbon dioxide coming in, glucose coming out, okay? In the mitochondria down here, we have oxygen coming in, water coming out, and ATP being released. So the question says, which statement describes how photosynthesis and cellular respiration are interrelated? So which of these is correct? So B is the answer that's chosen here. So I'm just going to go through real quick. Oxygen. A, oxygen is produced during cellular respiration and stored during photosynthesis is not correct, because in this picture you can see the oxygen is not being released during respiration, it's being added. So that one's a definite no. C, photosynthesis releases the energy that is stored during the process of cellular respiration. It releases glucose, but doesn't necessarily release something to be stored. So that one would be, if that was going to be one, that would be questionable. D, glucose is used during cellular respiration to produce food that is broken down during photosynthesis. Again, that's not going to be correct, because even though glucose does go in to cellular respiration, it's not producing food, it's producing ATP. So D is a definite no. So it would be back between the B and C, and based on what we are given here, B is our definite best answer. So excellent job on that. Carbon dioxide and water are both released by our cellular respiration. They are both products here, and they are both used in our photosynthesis they're going into the chloroplast there. So they're coming, the arrow comes off the mitochondria, showing you that those are both released, and then the arrow goes from them to the chloroplast, showing that that's where they're used. So they are released from the cellular respiration and used in photosynthesis. Excellent job. So recap, these are the two basic reactions of photosynthesis and respiration. In photosynthesis, you have the carbon dioxide plus the water plus the light energy, giving us glucose and oxygen as a byproduct, whereas in respiration, we have the glucose and the oxygen that then get converted into releasing carbon dioxide, water, and then ATP energy. So they're opposites of one another, basically. And it's important to remember that photosynthesis happens in the chloroplasts, which are only found in autotrophic cells, mostly plant cells, but some algae or single-celled organisms. And mitochondria are one of the main areas where respiration takes place. Excuse me. Although they also takes place partly in the cytoplasm as well. But this happens not just in animals, but also in plants. So that's something to keep in mind. I know there's a lot of misconceptions. Sometimes people will think, well, photosynthesis is only in plants, respiration is only in animals, and that's not really correct. Photosynthesis is only in autotrophs or plants, but respiration is in both. Respiration is in all eukaryotic organisms because they all have to convert the glucose into the ATP for the cells to do work. So that's something to remember, okay? All right, I think this is the last question to go over. So we have water as our last main topic with the biochemistry. Water is essential for life. Its special properties make water the single most important molecule in plant life. Which of the following properties of water enables it to move from the roots to the leaves of plants? You got a fast answer here. Excellent. Water exhibits cohesive behavior, and that is correct. Very good. Water does expand as it freezes, so that is a correct statement, but it's not correct answer for this question because it's specifically asking for the property that allows water to move from the roots to the leaves of the plant. So even though water expanding as it freezes is a correct statement, it's not correct for the question being asked. Same thing with B. Water is an excellent solvent. Again, that is correct, but it doesn't help the water move from the roots to the leaves. And then D, water is able to moderate temperatures. Again, another correct statement about water. It is another property of water, but not one that allows the water to move from roots to leaves. So this is one of those questions where all of the answers do have a correctness to them as far as what kind of properties water has, but the key is making sure that you're answering specifically to what the question's asking about. In this case, is wanting to know about that water movement in the plants. Okay, so this is just a quick slide reviewing some of those main properties of water that are important to remember. Water is one of those unique molecules with its polar nature because of the oxygen being slightly negative and the hydrogen being slightly positive that allows it to bond to itself as well as to other things very easily. That's what leads to the cohesion and adhesion, which forms the surface tension, as well as allowing the molecule to move upwards against gravity in those plants. It also helps water be very good at moderation in temperature. It has a high heat capacity, which is one of the reasons why when it's 100 degrees out here in Florida, we don't necessarily see our pools boiling or the lakes boiling because it takes a lot longer for our water temperatures to increase to the same temperature as the air temperature. So even though right now we're hitting the 80s, some of the pools and stuff are still not quite warm enough to go swimming in because the water hasn't quite gotten there. It is the universal solvent. Again, that polarity, that positive and negative side to the molecule allows it to attract different ions and different parts of other molecules, which is what helps break them down very easily, and that's why it's called the universal solvent and dissolves things very easily. And then it is one of the few compounds that actually, substances that will expand when it becomes a solid. Most substances, when they become a solid, condense and become more dense and would sink, but because of the polarity of the molecule, it actually expands, and that makes it less dense, so that is why ice floats. It was one of the few substances that does that. All right, and that is it. Does anybody have any questions or anything? I just wanted to, the whole point of this was to give you an idea of what some of the questions are going to look like and just a quick review of some of the information that goes along with why the questions are answered the way they are. So we'll do this every week following Ms. Tyson's content. So any questions or any comments? All right. Well, thank you guys for joining me, and look forward to hopefully seeing you back here again next Thursday, okay? Have a wonderful day.